Hi, we're group TMD6.
Our presentation is about gene therapy, which uses genes to prevent or treat diseases. Different diseases that gene therapy can help solve include liver cancer, ovary cancer, prostate cancer, and colorectal cancer.
Currently, cationic lipids, liposomes, and nanoemulsions in gene therapy have not proven to be efficient in taking genes into the patient's body. To ensure that diseases such as my partner have mentioned are cured, our focus is to find the optimal conditions to maximize genes going into the body. So essentially, our hypothesis covered the optimal conditions of a volume to weight ratio, duration time periods, types of vectors, and interference. For each of the optimal conditions mentioned previously, we conducted a separate trial. For this trial, we first had a sample in the first row and a backdrop TE buffer. Then we had a trident buffer, and the trident buffer was used to lyse the cells to get not only the total mRNA, but also the encapsulated mRNA. The ribogreen was used to intercalate with the mRNA to fluoresce and use the plate reader to measure and quantify the mRNA concentrations. To our first experiment, we looked at the volume to weight ratios, and we found that 50 nanograms of mRNA to 50 microliters of LFMM was the most optimal. This optimal condition led to a significantly increased encapsulation efficiency, specifically around 33%, 64% increase in encapsulation efficiency. Hence, this found a concrete result of the optimal volume to weight ratio to optimize encapsulation efficiencies. Next, we looked at any interference that might be available or present in the experiment itself, and we found two interference, trident and optimum, and these are found in laboratory settings, and we found that these significantly hindered encapsulation efficiencies. For example, the removal of these interference led to a 25% increase in the encapsulation efficiencies. Hence, we identified novel interference that are present in laboratories that could hinder encapsulation efficiencies and hinder gene therapy success. Finally, we looked at the optimal vector and also duration time periods in which the vector was most optimal for encapsulation efficiency. We found that LFMM was significantly better than LNP, or the liquid reagent of LFMM was better than the lipid nanoparticles of LNP. In addition, we found the specific optimal duration time period, which was 1 to 6 days, and past this date, the encapsulation efficiency went below 70%, showing that gene therapy would not be successful. Hence, we quantified and found concrete results of optimal conditions, including the volume to weight ratio, duration time periods, and the significance of interference that could impact researchers in gene therapy today.
Our study has found concrete research that can increase encapsulation efficiencies up to 64%. Using this, we can help millions of people with diseases like cystic fibrosis, heart disease, and AIDS.
In a world where conventional treatments are unable to find a solution, our study is one stepping stone in making gene therapy a game changer.